Continuing on with our tour of the cells, we've made it into the inside of prokaryotic cells in part 5 of the video, and in part 6 we're going to take a look inside of our eukaryotic cells that are out there. And when we look at our eukaryotic cells, again, you can find all these organelles inside of there, these little structures. And a few of them do not have membranes around them, and also a few of them do have membranes. And so we'll take a look at some of these non-membrane bound structures first, and then move on to look at the membrane bound organelles. And one of the things that's really hard to draw in this is there's actually a cell skeleton that spans throughout the cells that we can see a little bit more effectively when you look at these fluorescently labeled photomicrographs right here. So here you've painted the cells with fluorescent chemicals that will stick to different parts. And the little blue thing, that's actually where the chromosomes are inside of the cells. And the green thing that we see highlighted here are some protein fibers known as microtubules. The red thing that we see here is other protein fibers known as microfilaments. And in this figure, we can see what are known as intermediate filaments. So there's 3 different versions of bones, really, if you will, that make up the cytoskeleton inside of the cells that are out there. And researchers have actually found that there are related forms of all 3 of these types of protein fibers in archaea cells. And so remember that idea of phylogeny, that eukaryotic cells are descendants of these ancient archaea cells. And so the fact that they have something that is similar to these is really sort of nature's evolution, that eukaryotic cells have taken what the archaea cells started out with and remodeled them and made them a little bit more effective for their own use out there compared to the archaea ancestors. If we look at each of these fibers individually, again, the microtubules over here that we see, they're made of little proteins called tubulins that form these hollow tubes right here. So imagine taking, think like a soda can and making stacks of them to form this helix where you've got this hollow core inside of there. And when you look at the function for these guys, you can see they're extending throughout the cytoplasm for the cell, but you've definitely got these almost networks of these tubulin proteins making up these microtubules. And so they're going to be involved in moving things throughout the cytoplasm. And here we can see an example of a sac that is being carried along this microtubule by something that's known as a motor protein. And so it's basically using this as almost like a road, and the motor protein is dragging that sac along the road that extends throughout the cell there. When we look at our microfilaments, these guys are going to be like long, thin threads, and they're basically stacks of a protein known as actin right here. So this is a long chain of actin proteins that are sort of wrapped around each other to make this microfilament-like structure. These guys are going to be involved in a variety of cellular processes, and they're really easy because you can add actin proteins onto the end of this, and if you want to make this shorter, you just need to chop these guys off on the end there. So actin filaments are very dynamic and can change their length relatively quickly, and they think that these are going to be involved in things like if you see an amoeba moving around, it can extend out its cytoplasmic membrane because actin proteins in the microfilaments are pushing on the membrane or pulling it back there. They're also going to be involved in cell division, in basically forming what's known as a contractile ring. So imagine taking like a zip tie around a bag, and if you use that zip tie and you tighten it, you can separate that bag into 2 little sacs. Well, that's what cells are doing when they're dividing there. So really some interesting kinds of things going on with our actin proteins out there that are a little bit different from the microtubules. The final kind of thing that we can find out here when we look at these, and here's some figures just showing you different things that the actin proteins can do in the microfilaments there. But when we look at our intermediate filaments, these are kind of interesting. They are made of a variety of different proteins here that are going to fold together, including some of that keratin again. And these tend to be a little bit more permanent structures than what we find with the other cytoskeletal elements that we've seen right there. And when you look at this, you can see this is actually a picture showing you that these intermediate filaments right here are actually coming in or holding the cell membrane really close to this other cell right here. And this is going to allow these cells to come in contact right there and form something that's known as a desmosome. So this is really a tight junction between these cells that's being held together by these intermediate filaments. And if you look at this figure on the right, you can actually see the nucleus where you've got this nice bright green stain around here that is these intermediate filaments that are forming this little ring right here. And so if you look closely at this little cartoon, you can sort of see the intermediate filaments are making up this inner layer inside of this structure. So it's almost like they're holding this nucleus together right there. So really kind of interesting when you look at this thing, because it is going to be involved in, again, these more permanent structures that are out there. There is a modification of the microtubules out there, and they can form these little barrel-like structures right here. And each of these things is what's known as a centriole. And so these microtubules, again, those hollow tubes that we saw before, are lining up, and you can see this little bundle of 3 of them right there. And so they're going to make this regular structure with these long tubes right here. And in animal cells, they're going to form what's known as a centrosome, where you've got 2 of these things that are positioned at right angles to each other. And what these guys do is they serve sort of as the foundation to build up more of these microtubule-like structures. And in animal cells, these centrosomes are going to be used to form this complex where you're actually moving your chromosomes around during cell division, like mitosis or meiosis, and you form what's known as the mitotic spindle. So you get these centrosomes to move to opposite sides of the cell, and you send out your microtubule tethers that latch onto the chromosome, and then they're going to drag the chromosomes to opposite sides of the cell when the cell is dividing there. Another related kind of structure that we can see out there, and again, you can see those stacks with these 3 microtubule barrels, or not barrel, these tubes that we see right here are arranged, and they come with 9 in this ring around the outside, and then 2 of those little tubes on the inside. And this is going to make up a structure that we find in our eukaryotic cells that's going to extend out to make your flagella and your cilia. And the interesting kind of thing when you look at this is we saw that our bacteria flagella were actually on the exterior of the cell. But if you look closely at an electron micrograph like this thing, the membrane actually extends around these things. So they actually are considered more like an internal structure right here. And what goes on with these things, it's really kind of interesting, is the cell is going to pull on the microtubules on one side, and that causes your flagella or your cilia to whip in this direction. And then they loosen that thing up, and it causes this thing to whip in this direction here. So effectively, these structures are going to whip back and forth. And that's going to push the cell, if you're looking at a flagella right there. With our bacteria, remember, those guys rotated around the hook. We still have this basal body, but our basal body for our eukaryotic cells is really just these microtubules. And some people have hypothesized that when you look at the flagella or the cilia, they're basically those centrioles that have been repurposed for another thing to allow the cells to move around and do that taxis. And with cilia, they tend to be more numerous around the outside of the cells, and they think not only are they used in locomotion with something like a paramecium cell, but they can also be used to help the cell feed by churning up the water and possibly helping to move things into the mouth of the cell so they can consume it by something like phagocytosis. So again, some really interesting kind of structures, but no membranes are around these things. And the last thing to remember is that idea of the ribosomes that I talked about before, that in our eukaryotic cells, the ribosomes are 80S. They're the larger version of it. If we look at these membrane-bound things over here, we don't need that ribosome, so I'm going to take that little guy off of there. But you can see there's a variety of different membrane-bound organelles here. And so when we look at this, these different structures are going to have different functions inside of the cells. And so you can almost think of these things as workbenches. And you have a workbench where you do this little thing over here, and then this workbench we're going to process chemicals. This workbench we're going to do something else. And so that's what these organelles are being used for inside of our eukaryotic cells that are out there. We'll start out with the brains of the operation, the nucleus. And you can see in this little cartoon, it's based on looking at this transmission electron micrograph. So again, remember, TEMs are thin slices through the interior of the cell, and you shoot your electrons through it to visualize the different kinds of structures that are out there, but now you're down at that nanometer range. And when you look at the nucleus, kind of the interesting thing is there's actually two layers of membranes that surround the nucleus to make what's known as the nuclear envelope right there. And when you look at the nuclear envelope, things need to travel from the cytoplasm to get into the nucleus, but also the nucleus is sending things to the outside. And so built into this structure are these little things known as nuclear pores. So just like we had protein channels on the cytoplasmic membrane, you're also going to have protein channels in this nuclear envelope that are going to allow the transport or movement of molecules in and out of the nucleus there. If we also look inside the nucleus, we can find something that is known as chromatin. And this is where eukaryotic cells have linear chromosomes, not circular like what we saw with our prokaryotic cells, but linear chromosomes that are these DNA fibers that are wrapped around your histone proteins to form that chromatin. And this is going to be loosely wound inside of cells as they're normally doing their activities. But when eukaryotic cells are going through mitosis or meiosis, that's where your chromatin fibers wind up really tight. So you can think of it like a ball of yarn. When you're using it, you unwind it. And when you want to move it around, you wind it up really tight and form these tight chromatin fibers that we are more familiar with in terms of that picture of a chromosome in a eukaryotic cell. We can also find inside the nucleus this weird darkly staining kind of structure, and this is what's known as the nucleolus. And this is the site where you actually get your ribosomes assembled. So you're going to make your RNA molecules using your instructions in the DNA and then assemble the proteins on them in this region right here. And then they're going to get shipped out through the nuclear pores so they can float around in the cytoplasm where your ribosomes can do protein synthesis. Not all the ribosomes are going to go out to the cytoplasm there. And this is where we see this really interesting structure that is known as the endoplasmic reticulum. And endo is inside and plasmic is the fluid, so this is going to extend throughout the fluid inside of the cells, and it's actually attached to that nuclear envelope when we look at this endoplasmic reticulum. So just think of that membrane structure that is right there starting to fold out further and further. And some of those ribosomes that were assembled in the nucleolus are going to get transported to this, and they're going to get embedded into this membrane here. And this makes what's known as the rough endoplasmic reticulum. So this is where you've actually got ribosomes that are making proteins. The other side of this structure is what's known as the smooth endoplasmic reticulum. And this is going to be involved in producing lipids and other kinds of molecules that don't want to be out in the watery environment. So things like cholesterols and phospholipids to make more membranes are actually going to be assembled in the smooth endoplasmic reticulum. They've also implicated the SER in breaking down different kinds of drugs that are out there. So some cells that are exposed to a toxic environment are going to have a much larger size to their smooth ER compared to other cells because they're breaking down more of these toxins that are out there and detoxifying them. Typically found close to the endoplasmic reticulum is something that is known as the Golgi apparatus, or the Golgi complex, that was originally identified by an Italian who was staining cells with silver, and he found this weird kind of cup-like shaped thing that turns out to be just stacks of membrane discs that are assembled together right here. And this is separate from the endoplasmic reticulum, which, remember, is attached to your nucleus over here. And what goes on with this is the ER makes little sacs of materials there, and then it ships them out along your cytoskeleton. So again, there's your microtubules, and those little sacs of membrane that are filled with something are actually going to fuse with the membrane here and dump their contents. And inside of this Golgi body, this is where you're going to modify things like lipids and proteins, so you could turn them into glycolipids or glycoproteins. So add some sugars onto them and modify those things inside of there, and then you pinch off a little sac on the other side, and this vesicle is going to attach to your microtubules, where it will get shipped throughout the cell. And oftentimes these will go out to the cell membrane where they're going to be inserted, and that's how you can get some of those glycolipids that are found on the surface of your cells out there. They're actually being processed in the Golgi apparatus. When we look, we can also find other kinds of membrane-bound sacs inside of cells. And again, this depends on the type of cell that you're looking at. Animal cells are going to have lysosomes, which are basically a sac that is full of digestive enzymes, and they will use this to break down food and also break down organelles that are old and need to get recycled out there. You can also find peroxisomes, which are sacs that are full of enzymes that are going to play a role in making lipids, but also breaking down various molecules, including things like hydrogen peroxide. And so that's where we get this name peroxisome, because it has enzymes to break down peroxides that are out there. Another kind of membrane-bound sac that we can find inside of plant cells is the central vacuole. And this is where you're going to have these cells storing up a whole bunch of water and other kind of waste products. And we can also find this in some of the algae and protozoan cells that are out there. So again, when you look at your eukaryotic cells, you're looking at both non-membrane bound organelles, but also these membrane bound sacs to make up the cells.